Swayam Prabha. Digital India. Educated India.
Hello everyone, we are going to continue with our course Precision Oncology with another session, the Hallmarks of Cancer, the part 2. In the earlier class, we have really discussed about the hallmarks of cancer. Exactly, we have defined what exactly our hallmarks are, and then we went on to say what particular trait will definitely lead a normal cell to a cancer cell and thereby leading towards a progression of tumor. So, we have identified 8. Of course, we did not do it. Johann and Robert Weinberg have done it, and we were discussing about their 8. But there are another 2 more which are the vital hallmarks of cancer. We have completed discussing the sustaining proliferation, proliferating signaling, then how do cancers evade growth, suppressors, and then how do their resist cell death by apoptosis. So, we have discussed the very important what are the growth factors for a carcinogenic that will induce the formation of a tumorigenesis. So, there are many lines of evidence that indicate that tumorigenesis is a multi-step process, and these steps reflect genetic alteration. So, basically cancer is a genetic disease which we have discussed, and these events they drive to the progression of normal human cells into highly malignant cells. So, many types of cancers are diagnosed in human population with age dependent incidence, implicating there are many there are 4 to 7 rate limiting stochastic events. So, pathological analysis of this number of organ sites with cancer lesions, they represent that there are many intermediate steps in the process through which cells evolve progressively. It is a very progressive stage from a normal cell stage to a pre-malignant stage into an invasive cancer. So, cancer cells have defects in their regulatory circuits that govern normal cell proliferation. This is all what we were discussing in the last class. Just to very much quickly recap where we have left, here sustaining proliferative signaling, then evading growth suppressors, then evading the replicative motif we discussed about telomeres, then we discussed about apoptosis. So, this is real hallmarks. Then we discussed about how in each of this particular hallmark we have we are going to give we have given examples of mechanisms, and now we will further see the different very important hallmarks, the sustained angiogenesis, the tumor invasion and metastasis. Now, what is angiogenesis? So, angiogenesis plays an important role in the development of cancer, since it allows for the delivery of oxygen, nutrients and growth factors as well as tumor dissemination into the distant organs. So, what happens during angiogenesis? There is development of vasculature. What is a vasculature? Basically, it is giving birth to new endothelial cells or vessels and their assembly into tubes. That is called, we call it as vasculogenesis. In addition to the sprouting, that is either this from the existing new vessels branch, that is called sprouting angiogenesis, or from the existing ones. So, normal vasculature becomes, so after this embryogenesis, when the organ is formed and all, usually there is no need for a new vasculature formation or new vessel formation per se. So, what happens? So, suppose in an adult, in case there need to be a wound healing, or for example in female reproductive cycling, angiogenesis is turned on, but it is only for a transient period, when then again it goes turned back. Whereas in tumor, what happens? The tumor cells will always keep the angiogenic switch on and keep it activated, and thereby the normal quiescent vasculature will continue to sprout new vessels that will help the sustain and expanding of the tumor cells, or will help in sustain the neoplastic growth. So now what is supposing if you inhibit this angiogenesis, it is an important strategy for the prevention of multiple solid tumors that depend on cutting or at least reducing the blood supply to the tumor microgenesis. So these drugs are important part of treatment for some of types of cancer. As a standalone therapy also, some tumor angiogenesis can arrest or halt growth but will not eliminate the, they may totally not eliminate the tumor. Therefore, anti-angiogenic drugs are always used in combination with another anti-cancer treatment method like chemotherapy that could lead to lead to being critical for optimum better or patient output. What is angiogenesis? Angiogenesis is a multi-step process which involves endothelial cells and angiogenesis includes initiation, migration, tube formation and differentiation. Here we can see. So basically the oxygen and the nutrients supplied by the vasculature are crucial for the cell function and survival. So that is well. So during organogenesis, the closeness is ensured by coordinated growth of vessels and parenchyma. Once a tissue is formed, the growth of new blood vessels, the process of angiogenesis is transitory. It is not like every time you would need it, as I mentioned before. So this so usually but when in an suppose if the proliferation is aberrant or altered in cases like in your cancer, so initially they may not they will lack the angiogenic ability, but further on, in order to progress for these tumors to progress to larger size, to incipient neoplasias, the angiogenic ability is retained. So what is this? The angiogenic factors, they contribute to production of endothelial cells, which creates the walls of an existing small blood vessels here capillary, and the release of these particular factors. So binding of these factors will go on to the surface receptors, and thereby there is secretion of enzymes such as your matrix metalloprotein that reduce the and destroys your extra cellular matrix here. So then which will further lead to invasion of the matrix division, EC proliferation, and then starting of the strings of new ECs, they will organize into hollow tubes such as these, and thereby they forming new networks for blood vessels. So as a result, your vasculogenesis will be forming, and here you will have recruitment of pericytes and further on maturation, then differentiation. So in this mechanisms of in blood vessel formation, you have something called as the sprouting angiogenesis, where to the less frequent neovascularization is like formation of new from the new bone marrow derived endothelial progenitor cells. So and then intussusceptive microvascular growth and vasculogenic mimicry. So what are in each of these mechanism? In the sprouting angiogenesis, this is a very well known angiogenesis promoting mechanism which is adapted by tumor cells to induce their own vascularization from pre-existing host capillaries. From the ECM, that is your extracellular matrix component cells, they induce their own vascularization. So from ECM, soluble factors and well defined steps, as I mentioned before, define sprouting angiogenesis. Now what happens in vessel intussusception is like the development of the intravascular growth mechanism of splitting of the pre-existing vessels into new vascular structures, like how we see here. This is first described in the modeling of lung capillaries. Then you have your vasculogenic mimicry, which refers to the ability of some of the tumor cells, some of the malignant cells to start de-differentiation process to adapt to multiple cellular phenotypes, including endothelial like properties. So these cells finally converge in de novo vasculogenic like networks composed of red blood cells that are able to contribute to circulation. So these are the different mechanisms which cancer cells adapt for their own blood vessel vasculogenesis formation. So during tumor vasculogenesis, endothelial EPCs are mobilized as I've shown, and recruited in response to several chemokines, cytokines and growth factors produced by this tumor and the stromal cells. These are also very important. Normal cases, the stromal cells won't produce any of this particular growth factors or cytokines. But whereas upon the stimulus of the tumor cells, upon the growth factors released by the tumor cells, the stromal cells in turn secrete all this particular growth factors which will induce angiogenesis. So, vasculogenesis is also not only involved in primary, or not only involved for the for giving the nutrients or the oxygen supply to the primary tumor growth, but this vasculogenesis is also important for its distant metagenesis and dissemination of the tumor cell mass. Vessel interruption or the intussusceptive microvascular growth, whatever we have seen, is the development intravascular growth mechanism consisting of splitting of the pre-existing cells in vessels into two vascular structures, as we have seen before. Here you can see that from the single thing you have the two vascular structures arising. So, now what are the pro-angiogenic mediators in tumor angiogenesis? So, this is very important fact, these things. You have the several growth factors, you have the small mediators, you have the matrix degrading enzymes, then you have the cytokines, we have the bioactive lipids. So, all this pro-angiogenic mediators work in the tumor angiogenesis, like your VEGFs. So, vegetative endothelial growth factors, I will talk about all this in detail. Some of them like your TGF and all we have already discussed. Then this particular growth factors and your small mediators like your histamine, your nitric oxide, all this, you know, they will also will work towards promoting the angiogenesis in the tumor cell. Your matrix degrading enzymes, if you recollect in the earlier slide where how we said, you know, how this particular angiogenesis is formed. So, you have the matrix metalloproteins which are very key factors, and then your heparinases, and then your importantly your cytokines like such as your IL-8s, your colony stimulating factor 1 and your active lipids. So, these are all important ones for the continued for your pro-angiogenic events. So, for a angiogenic for a vessel formation is to be required. So, all this particular this particular cofactors are important. So, activation of the angiogenic process involves the breakdown of extracellular matrix at different levels for subsequent endothelial cell invasion and tube formation, as I have shown before. Apart from the role of tumor cells as principal secretors of endothelial cell promoters, the interplay with other stromal cells such as your pericytes is also very important for neo-vessel stability. So, when exactly your angiogenic switch occurs prior to tumor formation in this 3 transgenic mouse models of tumorigenesis is demonstrated in this particular slide. So, how do we know that a particular factor is for a pro-angiogenic or anti-angiogenic? So, people have studied, several groups have used transgenic mice models, where you overexpress this particular oncogene or you knock out this particular oncogene. So, transgenic mice carrying this dominant oncogenes or knockouts of even your tumor suppressor genes, they are being widely used. Of course, not only for angiogenesis, even for other any other hallmarks or any other to identify a growth factor for its key role in the mechanism study of your in cancer biology. So, this transgenic mice are very important. So, now here they have done 3 transgenic. This is a very important, this is a very good study which I am representing here to highlight, to show how this angiogenesis switch is switched on and what state. So, they have done 3 transgenic mouse models, your islet cell carcinoma, your dermal fibrocarcinoma and epidermal squamous cell carcinoma. In all 3 models, there is extensive vascularization and ongoing angiogenesis has been apparent. Here if you see in all these 3 models, this particular angiogenesis is very much apparent in towards the end stage. But where exactly is it coming up? So, yes, so this in the particular the tag oncogene model, normal oncogene expressing in the normal oncogene expressing islets, hyperplastic islets populated by here you have the hyperplastic islets which are populated by the proliferating cells. Here you can see. So with the histologic hallmarks of your CIS is carcinoma in situ. So here you would see this at this particular stage, you can see that new blood vessel growth has been activated. So your angiogenic switch is on here, and then which are in the, so thereby, and then here this will continue into your solid tumor also. Now coming back to the second transgenic model, where the which is also a multi-stage tumorigenesis model, in which the bovine papilloma oncogene elect dermal fibrosarcomas. So they have been analyzed for the angiogenesis BP1 oncogenes in transgenic mice which carry the BP1 oncogenic genes. So normally the normal dermis is initially converted into the state of a mild fibroblastoma. So which is seen as local small accumulation of fibroblasts, right. So then, so it is here not much of angiogenesis, but here you can clearly see the angiogenesis coming up, and it's in which is marked by dense arrays of here in this stage you have there is increase of more number of fibroblast cells, but and at the same time hyperproliferation, and you see the angiogenesis in the subsequent stage. And further on, this is to the squamous with the emergence of squamous carcinoma, there is all increase in of your fibroblast, and there is again your increase in of your angiogenesis. So even in the third model also, they have been able to show that this has been the basal keratinocytes were progressively transformed here in the normal squamous epithelium. Here you have the basal keratinoids which are progressively transformed into squamous cell carcinoma here by human HPV 16 oncogenes. You recollect back when we were discussing the basics of cancer, we were really highlighted how the HPV 16 is playing the role of an 16 can be the oncogene. Here in this particular here also you can clearly see that in the hyperplasia, then here also in the dysplasia. So just before progressing into the angiogenic switch is on in a at a very critical stage. So it not necessarily it is on only in the carcinoma stage, in the towards the end stage. In all the three models, if you see, so there is they have revealed that becomes activated during the early stages itself. So even in the early stages of the tumor development itself, the angiogenesis switch is on, and so thereby this is a very rate limiting step in the pathway to many solid tumors. So now we have seen in the mouse model. So now is it applicable in the human model? So for example, so here I have shown both, from the breast cancer, the breast ductal epithelium from the which arises from the ductal epithelium of the cell, and then the B is your normal cervical squamous here. Here what do you see? One is like can you see what happens in the mouse condition in the human condition also? Yes, here they have been able to show this particular group. So angiogenesis has been detected here also in the pre-neoplastic stage. So which here for the breast cancers which arise from the ductal epithelium of the breast. So so basically ductal lesions that are presented to be progenitors. So they are like suppose the moment there is increase in, there is an alteration, or there is a pathway which leads to aberrancy. So there is hyperplasia, dysplasia. Here you have the angiogenic carcinoma in situ, and then your invasive carcinoma. Here also this they have switched on angiogenesis, which you can clearly see by there is a lot of abundant new capillaries. So, this is an here also it is in the early stage, and whereas an angiogenic carcinoma in situ is an intermediate stage between carcinoma in situ and invasive cancer. So, this is also very important step here. So, now even in your normal cervical squamous epithelium, here you have carcinoma CIN1, CIN2 and then followed by your invasive carcinoma. So, what happens here? Here also there is an increase in a modest, a small increase in new vessel identity is evident in CIN. Here you see a small number, but whereas when it comes to CIN2, there is increase in angiogenesis or the new vessel densities. Here you can see it more. But whereas when it is invasive carcinoma, you see there is a lot of abundant vessels. So, thereby here you can see this angiogenic switch is from a vascular quiescence. So, there is no vascularization. So, here we call it as vascular quiescence, or your vascularization is in a quiet stage to a sustained neovascularization. So, we can clearly see how this transformation is happening. So, here this gives a complete nice model here, how the tumor cells, then you have your normal cells, and then you have your vascular cells, and then you have your immune cells, you know. So, this is a typical tumor microenvironment. So, which we will be discussing in later on in the further course. So, now how does all this will be will really try to see for the survival of this particular tumor cells. In the absence of new vasculature, suppose you do not need a new vasculature when the tumor growth is normally not more than 1 to 2 millimeter cube. So, the tumors will obtain nutrients from the nearby blood vessels and all that. But as this number is increasing and increasing, you know, beyond the cells grow beyond a restricted size, you know, they require this their vasculature network to be extended. So, this, you know, this angiogenic switch is what is clearly determines the tumor progression, and here they have come up with a balance hypothesis for this particular angiogenic disease. So, as I mentioned that there is the normally quiescent vasculature to sprout can be activated. So, you have the regular vasculature which is activated by your regular activators such as your FGF, your BFGF and your VEGF. So, by this that will change the relative balance of inducers. So, once they are activators, the inducers may keep the switch off. So, this inducers will go on to put on the angiogenic switch, and they will they will further see the process they will ensure the transition from the avascular stage to the angiogenic phases going on. So, and so this is the initial stage in the, this is a very good step in the, very preliminary step in the, for the tumor progression. So, you have again, you know, suppose you either reduce the inhibitor concentration, for example, your thrombospondin by loss of a tumor suppressor gene, or by increasing the activated levels, example for induction of your VEGF by hypoxia, can change the balance and activate this switch, leading to growth of new blood vessels. Thus, the angiogenic program is switched on in response to hypoxia, which together with the lack of nutrients. So, note two important factors, your hypoxia and nutrients. They promote the expression of inflammatory signals and cytokines that recruit the vascular cells for tumor vessel plexus formations. So, so as I mentioned, hypoxia triggers the transcription of several genes that are key mediators for angiogenic process such as your VEGF and your PGDF, your platelet derived growth factors. So, to continue further, you know, so for after once the angiogenesis is activated, tumor cells, they have to undergo numerous genetic and epigenetic rearrangements for them to grant to the angiogenic potential for both tumor growth and later metastasis. So, they have done a lot of experiments to show that lack of vascular network leads to tumor. So if there is no vascular network, there is the tumor, there is tumor apoptosis or necrosis, therefore thereby highlighting the importance of vasculature for tumor priming. So what happens, the angiogenic depends on the dynamic balance between your positive pro-angiogenic and negative anti-angiogenic factors, which we have seen before, which control vascular homeostasis. So it's very, very important to maintain the vascular homeostasis. So usually in the normal condition, the base is towards the balance is shifted towards negative regulation of angiogenic process. So thus maintaining the quiescence or the status quo of the vasculature. So now, so suppose during the first steps of tumorigenesis, what happens? High levels of strong angiogenic inducers such as VEGF and your FGF, they are released into the tumor ecosystem. VEGF is regarded as your regarded as the initiator and has been found to be expressed in many cancer types in response to different stimuli. Other than hypoxia or hypoglycemia and growth factors, and even there is the increase of your MIC also, it will increase your production of VEGF tenfold levels. So counterbalancing positive and negative signals. So this is how you have different pro-angiogenic factors, as I have mentioned before, which are coming up, which will aid in the increase in size of your tumor cell mass or to the neoplasia. So this angiogenesis initiated signals are exemplified by VEGF and acetic and basic, that is your fibroblast growth factors, that is your AB growth of FGF, which we have seen before. So each binds to the transmembrane, these in turn bind to your transmembrane tyrosine kinase receptors, which are displayed by the endothelial cells. So this is again a very important slide, which will show how is your angiogenic switch, is it in the on stage or is it in the off stage or in the on stage. In each of these arrows, the arrows, can you see the small arrows, they highlight the representative vessels. So in the quiet stage, you can see the, so for example in the mouse islets here, so mouse islets here, they show the full of proliferating beta cells. There is no absolutely or very quiet vasculature, so like a normal scenario. But whereas when it comes here, you can see that there is a new vessel growth. So here you can see the small arrows. So that is your with the hallmark experience. So this is a hallmark of vascular, there is appearance of vasculature here. So now here the human breast panels here, which shows a carcinoma in situ left, with here this is with the carcinoma in situ, and here in the normal duct associated vasculature there is nothing. But here, whereas when you see here, there is extensive neovascularization. Here can you see the arrows being pointed out. Coming here also again with the mouse dermis, which show prevascular it's in the prevascular, there mild fibromatosis, very mild, with very few vessels deep in the dermis. Whereas in the right, it's a very aggressive fibromatosis. Can you see here? With large capillaries here, they are spread throughout. Can you see the arrows? And they are thickened in the dermal layer. Coming here, is the human cervix, This is a very classic example here. So what is it? What does it happen here? The normal tissue has vessels deep in the stroma. Here can you see these vessels? Here can you see them? There it's in the very deep in the stroma. Whereas in the SYN3 lesion, as you can see, it has attracted, there is a dense arrays of new capillaries are distributed. This is an HPB16 induced epidermal dysplasia, where capillaries are distributed throughout the dermis. So just one quick slide on how VEGF that is a vascular endothelial growth factor that is secreted by tumor and stromal cells. It's also secreted by your endothelial cells and fibroblasts. It has multiple functions in the tumor microenvironment, which involve the ability of VEGF to interact with VEGF receptors that are expressed on different cell types, as I mentioned before. So, this acts as a primary stimulus, as I have been telling you in my last all 3-4 slides, for angiogenesis, which is the process, to stimulate the signaling pathways. This process of angiogenesis is the stimulate the signaling pathways that induce the proliferation and the migration of endothelial cells, and the ability, as you recollect in last 3-4 slides back, where I showed you one slide, these cells to degrade and remodel the extracellular matrix, that is your ECM. So, these processes culminate in sprouting angiogenesis and the formation of blood vessels. So, VEGF can also increase the vascular permeability, which results in the deposition of provisional fibrin matrix that triggers the formation of desmoplastic stroma. So, what does VEGF do? It also function as a chemoattractant to recruit Tregs, your T regulatory T cells, so that inhibit an anti-tumor immune response. So, then tumor fibroblasts also secrete, they also secrete. So, this goes in an autocrine fashion. So, it will first in turn trigger the fibroblast, and again they in turn secrete VEGF. So all this, again one more important factor is your neurophilin, that is expressed on tumor fibroblasts, may contribute to nuclear tumor growth. So, this again contributes to tumor growth by nucleating fibronectin fibril formation. But so this is how the VEGF targets the different, it will enable the vascular permeability. So, after seeing so much of tumor angiogenesis process, definitely there are inhibition strategies. So, it can be, because it is a complex angiogenesis process, it can be inhibited at different levels. For example, your direct vessel signaling inhibition approaches include your VEGF ligand inhibitors, your VEGF receptor inhibitors, which are important, and other growth factor inhibitors, that are released by the stromal or tumor cells. So, you even have your TKI inhibitors that block and also that block endothelial and pericyte cell activation, your tyrosine kinase inhibitors here. So, these are all, we have seen how this endothelial cell activation, pericyte activation, they are very crucial steps, for the process of angiogenesis to be completed. So, novel anti-angiogenic strategies are directed towards your EPC recruitment, your endothelial progenital cell recruitment inhibition, and so which occurs via your stromal derived factor or chemokine receptor type 4 signaling blockade. So, then again your extracellular matrix remodeling inhibition. So, considering all this contribution of your EPCs to tumor angiogenesis and metagenesis, so blocking of your EPC activation is a recently explored strategy for new blood vessel and metastatic abrogation or stopping or abortion, whatever you call it. This is how this is achieved. So, you are having different kind of inhibitors. Now, we move on to another very important hallmark pathway of cancer pathway, which is activating invasion and metastasis. So, first thing, let us come back to very important part, in the whole cancer biology, very important word is metastasis. So, first, the word metastasis was coined way back in 1829 by Jean-Claude. So, where he said that, the transfer of disease from one organ or part to another, but not directly connected to that thing. So if you see the whole process, so they came up with one particular hypothesis, So first thing, it's like if you see from 1830, so I am dividing this whole timeline into two slides. So from 1830 to 1959 or 55. So first how, I'll explain slowly how each term. So the first term was coined, then how this tumor first suggestion they came up in 1838, right, like how the tumor dissemination is determined by some mechanical. Then they came up with the seed and soil hypothesis for organs with disseminated cancer in 1899. So then slowly they went on to say that metastasis is here the first murine model of metastasis was developed in 1913, and then here if you see, the role of cellular adhesiveness in metastasis and distribution of embolic tumor cells was given in 1932. Then they said, see, it's a slowly stepwise, then how cells adapted to ascites growth persist in parent tumor have increased malignant phenotype. So this and then so whatever, it's been a slow understanding process of this very complex system. So what did they do first? In 1888, the English surgeon Stephen Paget, he identified the role of host tumor interactions and and he addressed the question, what is that decides, that organ shall suffer in case of disseminated cancer disease. So this he has given that. So then he concluded that remote organs cannot be altogether passive or indifferent regarding embolism. And so then he came up with a very principle called seed and soil principle, which states that when a plant goes to seed, the seeds are carried in all directions, but they can only live and grow if they fall on their own congenial soil. So 40 years later, somebody called Ewing, he challenged the seed and soil hypothesis. He proposed that, so 40 years later in 1928, Ewing challenged the seed and soil hypothesis. He proposed that the mechanical forces and circulatory patterns between the primary tumor and the secondary site they accounted for organ specificity. So although there's further on studies, by some Fiedler and co-workers, they showed that although tumor cells traffic through the vasculature of all organs, metastasis selectively develops in some congenial organs. So thus there is in the research in the early study 70s and 80s, they stimulated into the pathobiology. There's a lot of research in the pathobiology of metastasis. So thereby they came up with a term called niche or a local microenvironment, or now what we call tumor microenvironment for the primary tumor and the metastasized locali. Here during the 1970s and early 80s, they provided insight into the tumor biological heterogeneity and metastasis phenotype. So thus the outgrowth of metastatic lesion it develops a very important vascular network that evades, and this metastasis should be able to evade your host immune response and respond to organ specific patterns that influence their growth. One critical process in the whole thing is that, it is like the cells can invade the host trauma, penetrate blood vessels and enter the circulation to produce secondary metastasis again, the so called metastasis of metastasis, which we will see. Now here if you see, from the late 70s and 1980s, so as I said, there is the word niche which has been coined. So here from 1960s they have shown how the 1965 chromium labeled tumor cells used in dissemination studies. Then you have the how even if you are enzymatic manipulation of a cell surfaces, it affects your metastatic potential. Then lymphatic metastasis is distinguished from Hematogenous. So, there are two body fluids, or I am sure you all will agree, you have the lymph and the blood, right. So, how can, so they were able to distinguish here. Then here again, here in 1970s they have shown that metastasis can even come from survival of few tumor cells. So, this is a very very important issue in all the existing cancer, modern cancer treatment, where from the primary the metastasis may become more aggressive. So, even one breakthrough came in 1973, when human they were able to show that human tumors metastasis in thymic deficient nude mice. Here and then again they were able to show the in vivo selection of tumor cells for suppose they were able to select the in vivo cells for metastatic potential. So, this is again another breakthrough which came in 1973. And then a metastatic cascade was proposed for sequential events in disseminated cancer. So, then how is organ specificity, like for your lung, kidney, brain, breast, its specificity of metastasis is determined by your cell adhesion. So, then again the clonal evolution of tumor cell population was evolved, So, this also we will discuss in the coming slides, And then how it was shown that how invasion and metastasis are linked to metastatic cells which produce proteolytic enzymes. And then are all this metastatic neoplasm, are they clonal or are they heterogeneous? So, this also they were able to show that metastasis is an heterogeneous population. It is not like a single gene population. So, this is exactly what I was telling you. So, how you have your this hypothesis, they suggested that towards malignancy is a tumor progression towards malignancy is accompanied by heightened genetic instability of the progressing cells, which we all are aware. Come back to our first class, where we have shown the genetic instability, and then with your Darwin selection. Do you recollect that, only the survival of the fittest? Supposing the more mutations happen, there will be so the normal is preferred over the mutant, right. So, what happens? So, in the metastasis populations resistant to normal hemostatic growth factors, chemotherapy intervention and environmental restraint. So, all this metastatic population is resistant to all this particular normal signals. So, this is how it goes. So, here what happens is like so they measure the Siphon and Fiedler, they measure the mutation rates of paired metastatic and non-metastatic cloned lines isolated from different many murine tumors. So, they have found that highly metastatic cells are phenotypically less stable and than their benign counterparts. So, the mutation rate for the highly metastatic clones was higher, very very higher fold. So, the metastatic clones are very have a very high rate of mutations than the non-metastasized or poorly metastasized clones. So, this slides will give the basically, so it is a prelude to the previous slide. So, to the next slide. So, what are the steps in metastatic process? After the initial transforming event, the growth of neoplastic cells is progressive and frequently slow. So, what happens, as I mentioned, vascularization is required for tumor mass to excrete for a small from a small tumor cell size of 1 mm to 2 mm diameter. So, there is a lot of synthesis and secretion of angiogenesis factors that have role in establishing a vascular network within the surrounding host tissue. So, local invasion of the host stroma by tumor cells can occur by multiple mechanisms, including but not limited to thin walled venules and lymphatic vessels, both of which offer little resistance to tumor cell invasion. So, circulation of this emboli within the vascular, both hematologic and lymphatic, survival of tumor cells that traffic through the circulation and arrest in the capillary bed. So, then you have the extravasation. So, all these steps are very very important in the metastatic process. So, then once this vascularization is accomplished and the host defenses against host, I mean they are able to modulate the host immune response, there is again the reinitiation of these process of the development of metastasis from metastasis. So, the key change that occurs during cancer associated EMT or your epithelial mesenchymal transition is the reduced expression of or the loss of the E-catherine. So, this is a very, very important molecule. So, although there are multiple epithelial proteins, but which are down regulated in this is like E-catherine is reduced expression. So, this is like then as I explained in the previous slide. So, this is all the different steps of the metastasis. After the initial transforming event, the growth of is progressive. So, now we can see that there is the secretion of after the secretion of angiogenesis factors that have a critical role in establishing a vascular network with the host surrounding tissue. So, the local invasion of the host stroma by tumor cells can occur by multiple mechanisms. So, like how we can see here, like so detachment and embolism of this particular aggregates, tumor cell aggregates may be increased in size via interaction with hemopoietic cells with the circulation. So, circulation of these emboli within the vascular, both hematologic and lymphatic. So, now survival of the tumor cells that are trafficked. So, how are they? Once they are trafficked into the circulation through, through your both your lymphatic and the hematologic circulation system, you know. So, once they are doing, they are ready to metastasize in other organs. As mentioned in the previous slide, this is how the whole sequential process of metastasis is. So, once the tumor mass exceeds 1 to 2 mm², there is angiogenesis and then there is the motility and invasion. So, here the key change that occurs during cancer associated, one of the key factor is your EMT, epithelial mesenchymal transition, and this is evidenced by reduced expression or loss of your E-catherine protein along with other many epithelial proteins which are also down regulated. So, now after this particular multicell aggregates, you know, there is a lot of then you have your vascularization. So, there is transport of this particular aggregates, that is your emboli of your tumor cell emboli to through the blood circulation or through your lymphatic system, you know, and then again they go into and penetrate into the into organ parenchyma. So, basically they adhere in other particular organs, and there they respond to tumor microenvironment. Then again another tumor cell proliferation and angiogenesis occurs, and then again metastasis. So, once again they have established here, it happens a metastasis of metastasis. So, another very important hallmark which we are going to go another is like, you know, a tumor promoting inflammation. So, this we will be discussing in our next session, that is your molecular basis of carcinogenic. So, where I will talk in detail about the inflammations. So, what happens is, this is how you know, the white blood cells in tumors, they provide the first clue that inflammation is linked to cancer. And there is a lot of lymphoreticular infiltrate which has reflected the origin of cancer at sites of chronic inflammation. So, tumors can secrete growth factors and cytokines that result in the expansion and mobilization of your myeloid progenitors from the marrow, with trafficking to various extramedullary sites including your spleen, liver, lungs, and primary and metastatic tumor lesions. So, these committed myeloid progenitors, or what we call them as your CMPs, they can mature either into your dendritic cells, myeloid derived suppressor cells and macrophages, including your tumor associated macrophages, and they become activated or paralyzed within the tumor microenvironment. So, these heterogeneous cells include your so there will be a complete of heterogeneous cells which include progenitor cells, immature cells, mature and activated cells. So, depending upon the infiltrate substrate and the extent of maturation and activation, these cells can be a critical component and regulator of your vascularigenesis and angiogenesis. And suppose what and then there is tumor uh, infiltration or tumor growth, or sometimes it can even be a tumor regression or no growth. So, tumor is something what earlier they postulated that it could be a wound that does not heal. So, we should really be able to show that can a wound heal or not, a wound not healing, can it, how does, so now what exactly a cancer cell does is it will modulate the wound healing mechanisms for its own proliferation or for its successful establishment. So basically a tumors hijack the wound healing repair programs. So how do you compare a chronic wound and a tumor wound? Here between, to be very frank, there are many similarities between a cancerous tumor and the process of wound healing here. So both here you can see how very well the top is your wound healing and here is a tumor. How both involve the growth and survival and migration of cells. Here you can see there is lot of recruitment of cells and they require new blood vessels. Both of them require new blood vessels. Now all these processes are controlled by growth factors and again signaling molecule. Please recollect your earlier slides. So, just as the immune cells gather near a site of injury here to secrete growth factors to benign for a tissue repair, tumors can also surround themselves with, so they also surround themselves with immune cells that secrete the same growth factors to control, to promote their uncontrolled growth. So, as I said, they hijack your wound repair mechanisms. So, what happens usually the wound healing, if you see, it is by several subsequential steps. After injury, what it happens here is which is referred to as inflammation or proliferation dissolution, and then you have your remodeling where it is completely healed. So, immune cells are playing a key regulator in wound repair program. So, in the neutrophils kill the microbes and so you have the macrophage factor phagocytizing the apoptotic neutrophils, while skin resident or infiltrating T cells like they produce IL-7 and then IL-22 TNFL to amplify the host defense mechanism. So, during your proliferation stage the macrophage switch to anti-inflammatory phenotype, that is your M-sub or M-sub macrophages and N-sub neutrophils. For tumor associated neutrophils, that is called your TANs. So, what TANs, Tregs and other immunocells may help to attenuate the, so here the inflammation response is attenuated and this will help in the resolution and tissue modeling. So, chronic wounds get trapped here, many, if it is a chronic wound they will get trapped in the inflammation phase, exacerbate and thus hinder. So, there will be there is a hindrance of tissue repair in the chronic wound healer. But tumors on the other hand, what do? They hijack at this particular proliferating and the remodeling program and provide signals that can create a continuous angiogenic and immunosuppressive environment here that enables to that here the angiogenic and the immunosuppress, what will it enable the tumors to? It escapes the immunosurveillance. So, tumor remain in the proliferative phase upon the onset of, after the onset of angiogenesis, they continue to divide and divide. So, therefore, it remains in the upon the onset of angiogenesis. Here please keep in mind tumors will remain in the proliferative stage here at this particular state. So, anti-angiogenic immunotherapies induce an inflammation program in tumors that reawakens and boosts an anti-tumor response. So, that therefore, the goal is to create a homeostatic situation in which tumor cells are eliminated and a normal tissue architecture is achieved. So, two new hallmarks were added in the next generation of hallmarks of cancers. So, one of them is the reprogramming of energy metabolism. So, one very key important factor which we will be discussing is your Warburg effect. So, which is an anomalous characteristic of cancer cell energy metabolism. So, what happens here in an aerobic conditions, normal cells first process glucose first into pyruvate via the glycosyls, and thereafter via the oxidative phosphorylation to carbon dioxide in the mitochondria. Under anaerobic conditions, glycolysis is favored and relatively little pyruvate is dispatched to the oxygen consuming mitochondria. So, in the scenarios of the chronic uncontrolled cell proliferation, which is the representative of neoplasia, so not only but there has to be adjustment of energy metabolism in order to promote cell growth and division. So, now as mentioned, normal cells process first into glucose, first to pyruvate via glycolysis in the cytosol, and therefore to and thereafter to carbon dioxide in the mitochondria. So, Warburg first observed an anomalous characteristic of cancer metabolism, that even in the presence of oxygen, cancer cells can reprogram their energy production. So, that is their glucose metabolism by limiting their energy production largely to glycolysis. So, thereby leading to a stage that has been termed aerobic glycolysis. So, please note even in the presence of oxygen, cancer cells can reprogram their glucose metabolism and thus their energy production, which is called aerobic glycolysis. So, the existence of this metabolic switch has been substantiated in further on by many experiments. Such reprogramming is that must compensate for their 18-fold lower efficiency of ATP production, which is efforted by the glycolysis relative to mitochondrial oxidative phosphorylation. So, this is this is called the Warburg effect. So, now what happens here is it's like so there you need to have there is the uptake of upregulating of glucose transporters such as your GLUT1, which increases the glucose import into the cytoplasm. So, increased uptake and have been documented in many tumor cell types. So, increased uptake and glucose. So, this is like in your PET CT, you know, there is something called 18-fluorodeoxyglucose, which is a reported dye. So, they make the patient which is a radio labeled analog of glucose. So, this is what is detected in your PET CT images, what we can clearly see. That is like the cancer cells have an increased uptake of glucose. So, wherever there is increased uptake in particular image, so they say that, for example, your liver there are mets, this is a lung, or from the breast lymph node they see that particular increase in the glucose uptake. So, this is all the evidence of your PET CTs. So, coming back, metabolism of cancer pathways is regulated by signaling pathways related to oncogenes and tumor suppressor genes again. So, P13K activates ATK. So, back to our pathway which stimulates glucose uptake, and the flux by directly controlling your glycolytic enzymes and activating your mTOR. So, then indirectly, and which causes metabolic changes by activating your HIF, hypoxia inducing factor protein, which activates PDK, which inactivates your mitochondrial pyruvate dehydrogenase complex, and thereby inhibits the entry of pyruvate into TCA cycle. So, it is a very interesting link of signaling pathways which is related to oncogenes and your tumor suppressor genes. So, basically what does it do? Basically metabolism of cancer cells is regulated by the signaling pathways which is related to the oncogenes and tumor suppressor genes, as we all know. So, here P13K activates AKT, which stimulates glucose uptake flux, which stimulates your glucose uptake flux, and flux by directly controlling the glycolytic enzymes and by activating mTOR. So, mTOR indirectly causes metabolic changes by activating HIF. HIF activates your PDK, which inactivates the mitochondrial pyruvate dehydrogenase complex and thereby inhibits the entry of pyruvate into your TCA cycle. P53 suppresses glycolysis by increasing the expression of TIGR, supporting the expression of your P10 and promoting oxidative phosphorylation by your SC2. So, this is like, so there is this increase in transcription of your glycolytic enzymes which and also involved in the glutamine metabolism. So, the whole there is altered metabolism to the transformation of normal cells to cancer cells, and it is believed to be this particular glucose metabolism alteration is conserved in most tumors. One more important hallmark here which we will really be discussing in the towards the end, that is your immunosurveillance or your immunotherapy. Here how cancer cells evade immune destruction. All through we have been talking about several, how the cancer cells gets over tumor cells, how do they evade. So, they will modulate their immune destruction mechanisms. So, or they evade the immune destruction mechanisms. So, tumor infiltrating immune cells are critical players in the tumor microenvironment which modulate the cancer cell functions. So, they are usually tumoral infiltrating are the immunocells, they are highly heterogenic, and plastic and may either suppress cancers or provide support for tumor growth. So, there are you have your tumor there is a wide range of studies have shown light on how tumor associated macrophages, they are one more important, very important factors, cells, your dendritic cells, your neutrophils, your mast cells, natural killer cells and lymphocytes contribute to the they all contribute to the extraction for the establishment of several hallmarks of cancer, but and they have become for the basis of successful cancer immunotherapy. But here if you can really see here, how the different cancer associated fibroblastic and your like each of this particular immune cells, they are contributing to different hallmarks of cancer, like your tumor associated vascular cells, your cancer associated fibroblastic cells, your infiltrating immune cells, like just come back to our first slide, how you have the reductionist view and a heterogenic view, So, how the each one is like now at the end of this particular session, it is not like, one particular event I will have only deregulating cellular energetics. So, only this hallmark altered, can it go for cancer? No. So, you have each one of this hallmark, playing in its own role, or it's just like the cancer cell will play through this particular hallmark or particular like inducing angiogenesis, which is a definite prerequisite, or it will resist cell death, like the program apoptosis. So, we have clearly seen, how does it resist all this. So, this particular all this, it particularly evades the immune destruction. So, this is also we will be seeing all this in particular details in the towards the end of our course. And now, so now we have studied so much about our hallmarks. Now, doesn't seem, is it in me, you have such, everything is so well defined, yeah. You know what are the growth suppressors, we know what are the immune, what are the proliferating signals. If now we know that where I have TKI inhibitors, now I am giving an EGFR. Chalo, everything is it such a simple journey? No. So, it is like a therapeutic targeting of the hallmarks of cancer is a very, very, very well approach for targeted therapy. So, for example, not particular, so now in the drug discovery programs, so now they are able to identify the drugs exactly. You have your kinases or you have your tyrosine kinase inhibitors, where exactly are they following. So, now like for example, your immune therapy, so you are having your immune activating anti-CTLA MA4 monoclonal antibodies. So, they particular target this. Then you have your telomerase inhibitors, which will target this particular hallmarks. So, then you have your anti-inflammatory drugs, then your inhibitors of VEGF signaling, these are all also given with another particular. So, for example, your PARP inhibitors, so which will serve to in the work towards the genomic instability and mutation. Then your aerobic glycolysis, like which deregulate cellular energetics. So, this drugs that interfere with each of the hallmark capabilities and hallmark enabling processes have been developed. So, there are many of these drugs, many of them are already in trials or in clinical testings, for they are putting into many, like for example your EGFR inhibitors, they are like your first generation, second generation, third generation EGFR inhibitors for lung cancer. So, there are been very much in treating in certain, they are helping in certain treating the certain forms of cancer. Thank you.